Here is an overview of the figure consisting of 4 panels. A wild type, no cancer, situation with unbound ligand. B, the same with bound ligand. C, a mutated KRAS protein with activated pathway, as happens in some lung cancers. And D, the same but treated with adagrasib. A closer look at the wild-type no-cancer situation. The KRAS, Kirsten rat sarcoma, pathway links a receptor called EGFR for epithelial growth factor receptor to cell proliferation through a veritable alphabet soup of intermediates. We can distinguish the activators from the effectors organized in 2 pathways, the MAPK and the phosphoinositide pathway. Regarding the activators, note the SOS protein, which has nothing to do with marine rescue and the Titanic, but stands for son of 7 less because of a homologous protein in Drosophila. SOS activates the guanosine exchange system, GES, which converts guanosine diphosphate, GDP, to guanosine triphosphate, GTP. In panel A, the ligand, for instance EGF, is not bound to the receptor and no cell proliferation occurs. When a ligand binds to the EGFR, SOS activates the guanosine exchange factors. In turn, inactive GDP-bound KRAS becomes activated through the replacement of GDP by guanosine triphosphate GTP. GTP-bound KRAS activates downstream signaling by binding to effector proteins such as BRAF to activate the mitogen-activated protein kinase MAPK and P110 to activate the phosphatidylinositol 3 kinase signaling cascades. The circle is closed by GAP, the GTPase activating protein, which hydrolyzes guanosine triphosphate to guanosine diphosphate. Panel C shows the effect of the G12C mutation carried by many tumors, in particular pulmonary adenocarcinomas. G12C means that in position 12, guanosine is replaced by cytosine, which causes a glycine to cysteine substitution in the KRAS protein. The mutated KRAS favors the binding of the guanosine exchange factors, leading to displacement of GDP and replacement by GTP without ligand binding. This ligand-independent signaling is oncogenic by increasing cell proliferation and survival and decreasing apoptosis. Panel D depicts the mode of action of adagrasib. The drug binds to the cysteine residue coded by the G12C mutation, locks the mutated KRAS into the inactive form, thus preventing the replacement of GDP by GTP and the downstream oncogenic signaling.